everyone. I'm Manju Sharma from Suresh Gyanivihar University, Jagatpura, Jaipur, from School of Agriculture. So, we are going to discuss about principles of plant pathology, in which we will talk about different types of organisms which cause the diseases in plants. So today we are going to discuss about eukaryotes and the disease which causing by eukaryotes. So in eukaryotes, basically protists, those are including in eukaryotes. Protists are unicellular, coinocytic or multicellular with little or no differentiation of cells and tissues. For example, fungi, which cause the disease that is wilt of cotton, protozoa, heart rot of coconut, and algae, which cause the disease red rust of mango. Plants, parasitic flowering plants or phindrogamic parasite, which cause broom rap of tobacco. Animals, multicellular, extensive differentiation of cells and tissues, for example, nematode and the disease which caused by nematode, that is root knot nematode. After that, mesobiotic agent. Mesobiotic in the sense which categorize between abiotic or biotic agent. Biotic or abiotic agent, that means a connection between living or non-organism living, that is called mesobiotic agents. So those have the capable of surviving in the dormant state for a relatively long period, usually between 3 and 15 years. That means the organism which if the host will be living, then they will be act as a living organism, and if the host is non-living, then they will be also act as a non-living organism. So, capable of surviving in the dormant state for a relatively long period, usually between 3 and 15 years. So, they include viruses and viroids. They are infectious agents. That means the disease caused by the viruses. So, the disease will be infectious. Infectious in the sense, whenever the plant which will be infected by some disease, then the another plant which is also comes in the contact of the infected plant, then they will be definitely infected by the same disease. And we can say that infection, that means there is the establishment of a pathogen between two organisms. So their multiplication in the living plants ensures that they are living, they can be crystallized and are considered non-living. Hence, they are called as mesobiotic agents. Viruses, for example, yellow mosaic of black gram, and viroids which cause the disease, that is spindle tuber of potato. So here we have seen the classification of disease on the basis of the type of cause via non-pathogenic or pathogenic. Biotic agent in which like phytoplasma, spiroplasma, virus, viroids, algae, protozoa, phindrogamic parasites or bacteria. So here the fastidious vascular bacteria RLB, we can say that rickettsia like bacteria, the RLB can be divided into three groups. Three groups like xylem limited RLB, phloem limited RLB, and non-tissue restricted. So first is the fungi and the disease causing by them. So here phytoplasma, which are like cell wall and are bounded by a unit membrane. So they are pleomorphic. They have fried egg appearance of colony. They are filterable through 450 nanometer membrane. They have both DNA and RNA. They cannot be grown on artificial media. They produce symptoms like little leaf, palloid spike, yellows, stunning, witches, broom, etc. So they are mostly transmitted by leaf hoppers. They are insensitive to penicillin or they are insensitive to penicillin and sensitive to tetracycline. Tetracycline, that means whenever we will give the treatment of tetracycline, then they will be show the sensitivity or the tetracycline which will be affected for the phytoplasma, but they will be insensitive to penicillin. That means, so for example, phalloidy of sesame, little leaf of brinjal. Like there is the appearance of phytoplasma. Then spiroplasma. Spiroplasma is helical, wall-less prokaryotes. They require cholesterol for growth and cause diseases in plants, insects, and rats. So they are insensitive to penicillin and sensitive to the erythrocin and tetracycline. For example, corn stunt or citrus stubborn disease. After that, virus. Viruses are ultra more microscopic nucleoprotein entities which are infectious agents and obligatory parasitic pathogen which are less than 200 micrometer in size or mu. So they are devoid of enzymes and depend on the host protein synthesis machinery for their replication or for their multiplication. So they have only one type of nucleic acid via RNA or DNA. So, most of the plant viruses, those having RNA, for example, TMV. So, few viruses contain DNA, example like cauliflower mosaic virus and banana bunchy top virus, maize streak virus, and sugar beet, and that is called the disease curly top virus. Then viroids. Viroids are small low molecular weight ribonucleic acid that can infect plant cells, replicate themselves, and cause disease. So, they are also called as mini viruses. Potato spindle tuber, chrysanthemum, stunt, coconut, etc. Then algae. So the study of algae is called phycology or algology. Algae are eukaryotic, unicellular or multicellular. Algae are eukaryotic, unicellular or multicellular organisms and mostly occur in aquatic environment. So many algae thrive as terrestrial or subterrestrial algae. So the size of algae ranges from 1 to many centimeters in length. So they contain chlorophyll and are photosynthesis. So they reproduce by asexual and sexual process. After that, protozoa. Protozoa, which belongs to the class Mastigophora, order Kenotoplastida, and fungi, that is trypnosomatide, have been known as parasites plants or parasitic plant. Protozoa attacking plants move by flagella. So, the mastigophora or flagellates are categorized by one or more long cylinder flagella and some or all stages of their life cycle. So, flagella are the organ by which the unicellular or the protozoans those can move easily. So the flagella used for locomotion and food capture, they are also used as sense organ. So the body of flagellates has a definite long oval a spherical form which is maintained by a thin flexible membrane cover. So, in some groups, it may be armored. Flagellates reproduce by longitudinal fusion. So, the flagellates apparently cause the phloem necrosis disease of coffee and the heart rot disease of coconut. So, the merchitase that is suppressive, sudden wilt of whiter, disease, or on the oil palm on the important disease in oil palm, and there is Phytomonas stele was described from sieve tubes of coconut and oil palm. After that, phindrogamic parasites. Phindrogamic parasites are flowering plants or seed plants which leads a pre, pre which lead a parasitic life on other living plants. So they Parasites a great number of economic plants and cause considerable loss in yield. The phindrogamic parasites involve or invade stem or root of the host. Some of these parasites possess chlorophyll 2, which manufacture carbohydrates to a limited extent and depends on the host for a mineral. So, the phindrogamic parasites. Phindrogamic parasites are flowering plants or seed plants which lead a parasitic life on other living plants. They parasitize a great number of economic plants and cause considerable loss in yield. The phindrogamic parasites invade stem or root of the host plants. Some of these parasites possess chlorophyll second, which manufacture carbohydrates to a limited extent and depend on the host for mineral, salts, and water. So these are generally called as semi or partial parasites. Some of the parasites which do not have chlorophyll second depend entirely on the host plants for their food material. They are called hollow or total parasite because if they are completely dependent or entirely dependent on the host, then they will be called hollow. And the some parasites those depends only for some things, like not entirely depend on their host, they are hemiparasites. So, nearly 2,500 species of phindrogamic parasites in 11 families have been recorded throughout the world. So, here the Lorienthaceae are a family of flowering plants. It consists of about 75 genera and thousand species of woody plants. One of the most unusual of all genera and species of Lorienthese is Tristerex aphyllus. So the misstall parasites two species of cactus, that is Trichoceros and Euclinia acidica. After that, bacteria. Bacteria are microscopic unicellular prokaryotes which lack chlorophyll. Second, these microorganisms are with a primitive nucleus lacking a clearly defined membrane, and basically the bacteria which lack of chlorophyll. So the bacteria are smaller than the fungi and measures about 0.5% to 1 and 2 to 5 micrometer in size. So more than 1600 bacterial species are known. Majority of them are saprophytes. Several species cause disease in human beings and animals. About 200 species of bacteria cause disease in plants. First report of plant disease by bacteria was made by T.J. Prull of the University of Illinois. So he showed that fire blight of apple and pear is caused by a bacterium, that is Irvinia. So bacteria have been defined by Clifton as extremely minute, rigid, essentially unicellular organisms free of true chlorophyll and generally devoid of any photosynthetic pigment. So most commonly multiplying actually by simple transverse fission, so the resulting cells being of equal or rarely equal size. Next is, what are the rickettsia like bacteria and why are they called so? Rickettsia like bacteria such as Escherichia and Coxiella are an unusual type of bacteria that cause several similar diseases. These bacteria differ from most other bacteria in that they can live and multiply by only inside the cells of another organism like host and cannot survive of other inside the cells of the another organism and cannot survive on their own in the environment. Next is fastidious vascular bacteria, that is RLB. Fastidious vascular bacteria are otherwise called rickettsia-like bacteria, rickettsia-like organisms, or fastidious prokaryotes, or rickettsia-like walled bacteria. So they are small bacteria with a cellular ultrastructure of typical gram-negative bacteria. They are very existing in their nutritional requirement, refusing to grow on routine bacteriological media. So, unlike molecules, MLO and spiroplasma, which do not have cell wall, RLB have a cell wall. So, MLO is restricted to phloem tissues, whereas RLB are restricted mostly to xylem or phloem. So, a common habit for both RLB and MLO is the insect body fluid, that is hemolymph. So, both the groups are dependent on insect vectors for transmission. Non tissue restricted RLB have also been observed in the plant diseases. So, they reproduce by binary fission. Mostly insect vectors transmit them. Penicillin is effective against RLB. Sulfa drug also inhibit them. So, the RLB can be divided into three groups like xylem limited RLB, phloem limited RLB, and non tissue restricted RLB. So, first is the xylem limited RLB. In general, xylem limited gram negative bacteria have elongated cells of 0.2 to 0.5 into 1.4 mu size, and it is by Davis et al. In 1981. So, the cells usually have wall defined cell wall and plasma membrane. Both are triple layered in structure. The wall are rigid or ribbed due to periodic infolding of the outer membrane of the wall. So these width of the ridges is about 45 to 75 nanometer. So in culture, the cells of Pierce disease, of grapevine and almond leaf scrotch are non-motile, gram negative, oxidase negative, and catalase positive. So they are susceptible to tetracycline but not to penicillin. That means whenever we will give the treatment of tetracycline, then they will be showing the susceptibility, and we can say that with the treatment of tetracycline, we can inhibit the growth of the xylem limited RLB but not by the penicillin. So, how there will be transmission of xylem limited bacteria? So, mostly through xylem feeding insects, like the insects, those feed in those will be feed on the xylem. Safe transmission and transmission through vegetative propagation have been reported. The insect vectors belongs to sharpshooter, leaf hoppers, and spittlebug or frog hoppers. So the RLB are not pathogenic to the vector. Then the Pierce disease of grapevine is spread by Homalacedisca, Congulata, Oncometopia, etc. So there is no incubation period in the body of the vector and infectively is lost after molting. Molting, that means molting which will be occur at a particular stage in which basically the animals those will be breaks down their outer covering or there will be some changes. So this is because the RLB accumulate only in the salivary syringe where they appear to attach in a polar orientation. So the transmission is accompanied by regurgitation of the bacteria into the xylem stream. So here the plant, infected plant, and there is the vector. So basically it will be feed on the xylem and by which they will be transmitted the disease. So here the glacier winged sharpshooter leaf hopper vector of Xylella fastidiosa by which it will be cause the disease, and these are the xylem limited bacteria, and these are the symptom or the symptoms after infection by Xylella fastidiosa. Then next is phloem limited RLB. So it is first recognized by D. Lafley and Alain Bove in 1970. 12 phloem restricted RLB have been identified. So RLB have not been cultured in vitro and Koch postulates proof. So therefore not much is known about their nature, taxonomy and serological relationship. Examples of phloem limited RLB include citrus greening, clover, club leaf, white clover disease, clover rugose, leaf curl, potato leaflet stunt, little leaf of Sida cordifolia and stunning of Dodder. These are the basically diseases which caused by the phloem limited bacteria. Symptoms like stunning, yellowing of young leaves, virescence of floral parts, premature death of the entire plant because the bacteria which will be direct feed on the phloem, that's why there will be the lack of nutrients. So directly it will be cause the death of the plant or premature death of the entire plant structure, like they are mostly rigid rods and gram negative and sensitive to the penicillin, like we can give the treatment of penicillin. So the cells measures 0.2 to 0.5 and 1 to 2 μm and are bound by a double membrane or a cell wall and cytoplasmic membrane. After that, transmission is by leaf opers, dodder, and grafting. Citrus greening is transmitted by citrus, Piaspella, and by vegetative propagation. Clover clubleaf multiples in its vector. Ageliopus novella. So the vector retains infectively throughout the vector retains infectivity throughout its life cycle, and the RLB is transovarially transmitted. And last one is like non-tissue restricted RLB. They are also found in parenchyma and meristematic cells of yellow of grapevine, chlorosis, and aspermy of wheat, apple proliferation, carrot proliferation, and necrosis of grapevine. So, a yellow of grapevine is transmitted by a nematode, that is Syphinema index. So, this kind, like these are the RLB in which we discuss about xylem limited bacteria or phloem limited bacteria. That means xylem limited, those will be directly invade the xylem tissue and then absorb the all like water content. And another type of bacteria are phloem limited bacteria which directly connected with the phloem tissue, so they will be directly absorb the nutrients, and that's why the plant will be died in premature. So this kind, the phloem limited or the xylem limited, which will be cause the disease in the plant. So firstly, the phloem limited RLB, which goes by recognized by D. Lafley and L. M. Bowe in 1970. There are 12 phloem restricted RLB have been identified. RLB have not been cultured in vitro and Koch postulates prove. Therefore, not much is known about their nature, taxonomy, and serological relationship. So here the examples of phloem limited RLB include citrus greening, clover club leaf, white clover disease, clover, rugose, leaf curl, potato leaflet stem, little leaf of Sida cordifolia, and stunning of dodder. So their symptoms are like stunning, yellowing of young leaves, virescence of floral parts, premature death of the entire plant. So their structure, like they are mostly rigid, roots and gram negative and sensitive to penicillin. So they cause measures. So the cells measure like 0.2 to 0.5, then 1 to 2 micrometer or mu-am and are bound by a double membrane or a cell wall and cytoplasmic membrane. Their transmission, like transmission is by leaf hoppers, dodo and grafting. Citrus greening is transmitted by citrus phyzilla and by vegetative propagation. Clover club leaf multiplies in its vector, that is Eukaryotes novella. So the vector retains infectivity throughout its life cycle, and the RLB is transferally transmitted. And these are the non-tissue restricted RLB. So basically, they are also found in parenchyma and meristematic cells of yellows of grapevine, chlorosis, and aspermi of wheat, apple, proliferation, carrot proliferation, and necrosis of grapevine. So, a yellow of grapevine is transmitted by a nematode, that is Ziphilema index, and this is all about the RLP. Thank you.